Hi, my name is Eilleen Falkenberry, and today I'm going to be talking about epigenetic regulators of rat brain development. So let's jump right in. So first off, we're going to give a little bit of background on what epigenetics actually is, discussing how it works, and then we're going to look at how it presents itself in plants and in rats. And that's going to lead us into the rationale, which is the link between environment and gene expression. And then our research question is basically what are the molecular causes of neurological development and how does epigenetics fit into this? Then we'll get into the hypotheses and predictions. We'll be looking at two different things. One of them is caspase-3. The other is DNA methylation in young mice. Then we'll get into some cool methods, which are really awesome, and then some interesting results. And then we'll conclude with what does this mean for us as people and what we can do for future directions. So just to orient everybody, I'm going to remind you all of the central dogma of biology, which is DNA gets transcribed into RNA, which gets translated into protein. And this is crucial for epigenetics because epigenetics is the regulation of those different points of the central dogma. Epigenetic processes are basically the environment regulating gene expression. It can happen at the DNA level all the way to the protein level. Some of the different types or different flavors of epigenetic regulation is DNA methylation, histone modifications, chromatin remodeling, and RNA regulation, which is something that I personally do. I do RNA interference, which is an epigenetic control at the RNA level. And these have impacts on a phenotype, which is how something is expressed. It can affect behavior, it can even affect cancer development, it can affect overall development. It's incredibly important to the well-being of an individual organism. So let's get into a little bit more of what DNA methylation and histone modification is. Let's start with the simple one, which is DNA methylation. That's when you have a teeny tiny little molecule, and whoops, it basically attaches itself to the DNA. You can see it over here on the left-hand side of the screen in the little red circles. It attaches upstream of a gene at the promoter. A promoter is when it's basically an area where a transcription factor can attach on and say, you know what, we're going to start gene transcription right now. So promoters are important, but if you add a methyl tag there, where those red circles are, it basically blocks transcription factors from attaching there. And then if a transcription factor cannot attach there, the gene cannot be transcribed and that gene is silenced, goes to sleep. Another type is histone modification. And a histone is a molecule composed of 4 different proteins, and they have little tails sticking out of them. I think they're made of lysine. And different molecules like acetyl groups or methyl groups, seen in the blue and red circles, can actually attach to these tails and they can cause a change. There are a couple of different changes that can happen. The one thing to note is that DNA, which is that blue line, actually wraps around a histone. It's a way of compacting it. And DNA can either be wrapped really tightly around or really loosely around the histone. This is important. If the DNA is wrapped super tightly around a histone, then RNA transcriptase can't get in there and read the gene. That gene is silenced. If it's wrapped very loosely, the transcriptase can get in there and it can read the gene. Now you can actually, or not you, but this can actually be changed by adding an acetyl group or methyl group, and it depends on what point of the tail you add it on. But it can actually cause the DNA to be wrapped more tightly around the histone or more loosely wrapped. And this can silence a gene or it can turn it on. Here's another cool picture of histones that kind of gives a little bit more meat into what's going on. You have the histone tail, DNA wrapping around it. And this is another way to think about this is you have a pencil, and a pencil can write and it can also erase things. So an epigenetic regulator can also write something or it can erase it. So if you have something like an acetyl group that'll turn a gene on, you're writing it. If you have a DNA methyl group that'll turn a gene off, you're erasing it. The important thing about a pencil, though, is you can always undo your action. These are undoable things. They're reversible. And that gets really important for plants, actually. Plants actually show a really cool phenotype when they're put under a lot of stress. So this graph is from Boyko, Alex et al., and they did a study about Arabidopsis, which is a mustard plant. And they subjected the F1, the parent generation, to lots of stressors, and like too much salt or too much heat or too much cold. It didn't kill the plant, but it did cause the methyl tags to be added to different genes. And they found that in the F2 generation, the progeny generation, there was actually more methylation. And they found that when there was more methylation, that the F2 generation was actually more tolerant of stress. But in the F3 generation, so the progeny of the F2 generation, did not show any of this. So the important thing is that epigenetic factors such as methylation can be inherited and it can be disinherited. So it's just like our pencil, writing and erasing, writing and erasing. And it can actually help animals cope with stress. But there's also a dark side of this. It can help them not cope with stress. So this is a very famous study about rat moms licking their pups. And when a rat mom licks their pups, it's considered nurturing, and that pup actually grows up to be very relaxed and less anxious. But this phenotype can actually be reversed. So when they injected the relaxed rat with methyl tags, it actually grew up to be an anxious rat. How did that happen? Well, what happened is you have something called glucocorticoids, and they help calm you down. And basically, the methyl tags had attached to the promoter of the glucocorticoid gene and effectively silenced it. So it couldn't get into the hippocampus and calm the rat down. So the rat was always stressed. And this is another form of epigenetic regulation that has a profound effect on an organism. So why is this important for brain development? Well, I think you can kind of guess, but there is a link already between environment, behavior, and gene regulation. So in the plants, we saw that if they were subjected to a lot of stress, then they tried to control their gene expression to be more resilient against that stress. That was their environment causing gene expression to change. Same thing happened to the rats. Their behavior changed when they were not just subjected to a stressful environment, but if they were injected with something that would turn off their ability to de-stress themselves. And that kind of leads us into our experiments, which are going to look at two different model organisms, rats and mice. Both are really good model organisms because humans share a lot of the same genes. But rats tend to be a more preferential model because their organs are bigger, they're smarter. You can use them for a lot of different studies, including how epigenetics affects the brain. So the big research question is, how does epigenetics affect rat brain development? And these are the two articles that we're going to be looking at. So how does it affect rat brain development? Oops, I don't know that. All right. Um, so here's what we know and what we don't know. We know that caspase-3, when knocked out, causes neuronal death. Caspase-3 is an important regulator of apoptosis. It also regulates cytoskeleton degradation, basically keeps everything in homeostasis, nice and happy medium. We also know that neurodivergent behavior can be induced, which means you place an organism in a specific type of environment, especially if there's not a lot of stimulation around it, that can actually lead to neurodivergent behaviors. Here's what we don't know. Don't know the regulatory mechanisms of caspase-3 in postnatal brain development and how it affects behavior. They suspected that it does, but they're not sure how. They also don't know the molecular mechanisms of how neurodivergent behavior can be induced or changed.